Okay, as I told you, there was going to be two parts to the muscle lecture. One was the directed teaching that you've already seen that was on the board. And this one is the PowerPoint, chapter 10, that I wanted you to download so you'd have it ready to go. I'm going to go over it. There's 62 slides. Some of them I'll go over a little quickly, but this should be a review. Okay, if you have any question, get a hold of me or the team, and we'll take care of it. Don't forget you have SI, and things can be answered there as well as in private tutoring. Okay, it says chapter 10, it says muscle tissue and sliding filament mechanism. Okay, we're going to talk about properties of muscle tissue. You already know this, okay? Contractibility, extensibility, that kind of stuff. Characters. Now, we had to pick one of the three muscle types. So I picked a skeletal muscle since that's the easiest one to identify, and it's the one that we've been talking about in lecture, okay? Because there's most of it. We'll talk about what is muscle. We'll talk about what is a muscle fiber, what's the name of a muscle cell, what's inside of a muscle cell, is there special names that go with muscles, cells, and that kind of stuff. Types of skeletal muscle fibers. Okay, this is the muscle, the fascicle, the myofiber, the myofibrils, and the myofilaments, actin and myosin. And that's the organization of it. We'll talk about the four-letter word exercise and skeletal muscle. We did this when we did, but would you guys leave me alone? Okay, we did this when we talked about bones, because we said the bone markings are caused from muscle pulling on the bones and moving it. You cannot increase your bone density by taking vitamins. You have to put a load on it, which means you have to exercise. And that'll keep you young, just like me. Levers and joint biomechanics. We're not going to talk about that, but just to kind of give you an idea, if a muscle is attached to the humerus and then it's attached to the head of the ulna, that muscle has to be very big in order to flex the arm. If it was attached to the wrist, it wouldn't have to be as strong because of the point of lever. Okay? All right. And let's see, what else do we have? Oh, I got more. The naming of skeletal muscles. I've been discussing this with the team. Just to have you do the illustrations would be a disservice. So we're looking into your online textbook, and there's a section on Atlas that has muscles, and we may go into that. I'll let you know at the beginning of the week how I'm going to construct that. But anyway, how you name a muscle, there's a variety of ways. Size, gluteus maximus, where it's located, rectus abdominis, rectus femoris. Talk about shape. They're named that way. They're named about what they connect and what they move and how they move. So we have, in the hands, we have flexors, extensors. Okay, so it's going to be named a whole bunch of different ways, not one just set way. Characteristics of cardiac and smooth. When I'm all done, I'll show the similarities and dissimilarities of cardiac and smooth to skeletal. Aging in the muscular system. That kind of goes back with that exercise stuff. Okay. Remember, and we've talked about this before, when you age, you don't make as many proteins like collagen. And since that goes down, since that is diminished, your strength and aging, your strength goes down and your aging obviously wears. Development of the muscular system, we're not going to talk about. Okay, here we go. 3 types of muscle tissue. This is a piece of cake, right? Skeletal, cardiac, smooth. Okay, am I going to ask you a question on that? Yeah, but that's low level. says there's about 700 skeletal muscles. When I did the lab introduction, I talked about there's about 700 muscles, and really there's 350, and then the 350 to put them back. So in other words, if these are flexor muscles, and there's 350 of them, you need 350 to put them back in the same position, original position. Okay, so those would be extensors. So anyway, that doesn't mean anything. Properties of muscle. There are 4 unique characteristics. Remember this one: excitability, contractibility, extensibility, and elasticity. Okay, I'll ask you a question on that on the exam. What this slide says, it says skeletal muscle is a tissue. Okay, skeletal muscle is a tissue, and you know that because it's muscle tissue, and it has all 4. What it means on this one, it has all 4 tissue types. It means in your muscle you have epithelium, muscle, nervous, and connective. Okay, so you have all 4 types, and that's what that means. Striated, remember that? A bands, I bands, the striations. You look for that in the microscope when we did skeletal muscle. Okay, there's skeletal muscle is striated and it's voluntary. Remember that? It's attached to bones, makes your skeleton move. It's voluntary. Okay, what does skeletal muscle do? Body movement. How does it do that? It's attached to bones and moves the bones at joints, and therefore the body moves. We have 206 bones in our body, and the reason for that is because we have 206 movements. And what I mean by movements is they move at that joint. So where 2 bones come together is a joint, and it's there because that allows for movement. Okay? If we only had 1 bone, you couldn't move. Maintenance and posture. Okay, when our muscles are not working, a certain percentage of them are constricted, and that gives us posture. I hope this doesn't happen to you, but more than likely it will. You're going to have to move somebody that is recently deceased. When you do that, the amount of strength you have seems like it's never going to be enough, even if the person only weighs 80 pounds, because there is no posture. Everything is loose. It's like trying to move Jello uphill. Anyway, maintenance of posture. Muscles do that. Temperature regulation. We talked about this. Every time a muscle contracts, whether it's a twitch or whether it's complete contraction, you use ATP to put it back. And when you do that, you burn ATP and it produces heat. So temperature regulation. When you're too cold, you may not know this, but your muscles are slightly contracting to burn ATP to create the heat. Storage and movement of materials. We know that we store glucose in muscle. We talked about that. Supports other tissues. I don't know what happened just now. Let me go back. It supports other structures. I guess that was it. Okay, now remember we said, what is a muscle composed of? So you've got a muscle in front of you and you're looking at it. What are you actually looking at? And if you take and look inside that muscle, you'll find out it's made of cylinders called fascicles. If you take one of those fascicles out and look inside of it, you're going to see that it contains other cylinders, and those other cylinders are actually muscle cells. And we know that a muscle cell's proper name is a myofiber, M-Y-O-F-I-B-E-R. You guys are driving me nuts. Okay. So then you see muscle fibers. This is going from smaller to larger. So the green is smaller. That is a muscle cell. You take a cluster of those, put them together, and you get a fascicle. So if this is a muscle cell, if this is a muscle cell and I put a bunch of these together, I will have a fascicle. If this is a fascicle, I put a bunch of these together, you'll have a muscle. Okay? Muscle fibers contain myofibrils. Okay, if this is a muscle cell, which is a myofiber, and I look inside, there'll be a bunch of horizontal, parallel proteins inside, or clusters of proteins called myofibrils. Myofibrils are composed of myofilaments, and we talked about the two most common myofilaments are actin and myosin. Okay, here's a picture. Let's see if I got the arrow. I don't know if the arrow is going to work in here. Nope, it only works over here. Well, that sucks. Oh, no, wait, here we go. Okay, humerus. Yes. Remember the coverings called mycelium? And there was another covering on the outside called fascia. So here's the mycelium. Maybe. Oh, there's the arrow. There's the arrow. See the mycelium right there? It happens to be epimycelium because it goes around the entire muscle. And then I said over the epimycelium is another coating we called fascia, which is like when you eat meat, it's the rubbery, the leathery type covering around the outside. You call it gristle, okay? Now, back here, these 1, 2, 3, 4, those are fascicles, which I showed you here. Those are fascicles. Inside the fascicles, 1, 2, 3, 4, all those, see all those? Those are muscle cells, myofibers. Now, if we could look inside, where'd my arrow go? Arrow. Now you're calling on the other phone, man. Okay, if we could look inside of there, there's little tiny tubes, and those are called myofibrils. If we look at those tubes and we could see inside those tubes, we'd end up seeing myofilaments, actin and myosin. So in this picture, what they show you is a muscle composed of fascicles. And here's a fascicle moved out, okay? And we see the fascicle has other circles inside, which are the actual cells, myofiber. If we look at the myofiber, now this is a muscle cell, we can see there's other little circles in it, and those are, there's clusters of those, and those are called myofibrils, okay? Which is what these things are. If we take out the myofibrils, see what they're showing here, right here, myofibrils. These are all myofibrils. Bunches of these together make up, or what's in a muscle cell. And then the myofilaments are actin and myosin. So what the thing shows you is, here's a fascicle taken out, okay? And this is what it would look like. And then you take the fascicle, and those are made of cells. Those are all cells, myofibers. And you take the cell out, and it is made up of myofibrils. If we went, and I have it coming up on another slide, inside of here is myofilaments, actin and myosin. Okay, and then this is kind of like a chart showing you the muscle, the fascicle, the myofiber, myofibril, dang it. The myofibril, someplace I got that arrow. There's a myofibril. And then the myofilaments, actin and myosin. Okay, good? All right, nothing new, nothing new. Mycelium, remember the 3 layers? The innermost one that goes around the cell, the myofiber, is endomycelium. The one that goes around the fascicle is perimycelium. And the one that goes completely around the muscle cell, the muscle itself, is called epimycelium. And then the last one is fascia. Okay, so anyway, here we go. Endomycelium tells you what it is, tells you what it's made out of. Perimycelium tells you where it's at, what it does and what it's made out of. And then there's the epimycelium that goes around the entire muscle. And then the extra coating around the outside is called fascia. Okay, and I put deep fascia, just fascia. Okay, fascia is all you need to know. We good? All right, here we go. Whoops, what was that one? Oh, I put superficial and deep. Don't worry about that. Muscle attachments. Okay. Remember we said that muscle does not attach to bone? Muscle becomes a tendon that attaches to bone. And you're saying to yourself, well, I didn't even think that tendons attach to bone. It attaches to periosteum. And you are correct. It attaches to, so when we talk about muscles attaching to bone, it's not correct. When we talk about tendons attaching to bone, we're going to loosely use that as being correct, because I want to talk about the difference between ligaments and tendons. But you know, it's actually the periosteum. So we have a tendon. And if you've ever eaten a tendon, it's really, really hard. Yes, you have. You gone to the kernel, and when you're chewing on a leg, okay, you're eating a drumstick, and you get all the way down to where it gets real small in diameter and it starts crunching. What you're crunching is cooked tendons, okay? Not bones, tendons. Boy, I'd like to have some of that right now. That sounds good, doesn't it? The kernel, original recipe. Ooh, ooh. Oh, that's right. It's got flour on it. That's not good. Okay, if we take a tendon, let's say a tendon's like my finger, it's round, okay? And we take and flatten it out like my hand. It's not called a tendon anymore. It's called an aponeurosis, okay? So an aponeurosis is a flat tendon. I'm going to put that on your exam. Okay, another terms. Remember origin, insertion, action, innervation. You should probably know that now, because if you started doing the lab, you had to look up and identify the muscle from that illustration. Remember, it was either a letter or a number. And then once you got that, then you had to take and go to the textbook, and in a table, it had origin, insertion, action, innervation. So there was 5 columns, one with the name of the muscle, one with the origin, one with the insertion, one with the action, one with the innervation. You're going to do that for all the muscles you identify. Okay, so it tells you about what is the, okay, I'm losing it here. Let me clear off some of these things. I shouldn't have 2 computers going. You know what it is? Most of this stuff is people just are bored, and they're just sending stupid messages out like, how are you? I don't want you to know how I am. Just get this stuff knocked out, okay? Okay. By the way, you know, between now and the end of the semester, there isn't any examinations. There's the online quizzes, and I sure in heck hope you're keeping up on those, okay? And after this presentation, I'm going to summarize what we've done this week. I'm not going to do it right here, okay? Okay, origin, the attachment site of a muscle that either doesn't move or moves only slightly. Insertion, the attachment site of a muscle that moves, period, moves, okay? Okay, they're using the example of the biceps muscle. Biceps brachii. Biceps means it has 2 points of origin. Okay, more people doing the same thing. Now they want to proctor the exams. They want you to have a log on and pay somebody to watch you. I told them, no, you can be trusted. Can't you? No, can't. I think you can. Okay. Muscle origin. Okay, if we look at this is the biceps brachii muscle. Remember, we're talking about fulcrums and biomechanics. Okay, if this muscle was down here, your muscle would not have to be as strong as it is if it's attached up here, because you've got to lever this whole hand, which has weight. Anyway, that's what we were talking about. Okay, you can see there's 2 points of origin. So when this muscle contracts, this part moves. Do you see it? This part doesn't move, or it moves very slightly. Origin, insertion. Are we good? Okay, now we have some specialized terms when we talk about muscles. Okay, first thing, myo muscle. Sarco, microscopic muscle. So as you can see right here, the plasma membrane is called a sarcolemma. It's still a plasma membrane, still a phospholipid bilayer. It still has all those things, still has protein gates in it, integral proteins, yada, 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 but it's called a sarcolemma. Got it? Sarcoplasm is the cytoplasm. So does it have organelles in it, cytosol and all that stuff that we learned? Yeah, it does. They just call it sarcoplasm. Are we good? Sarcoplasmic reticulum. Remember, I spent some time talking to you about this one. We have rough ER and smooth ER in all of our cells, including a muscle cell. But the smooth ER of a muscle cell is, by the way, you guys still have your pajamas on? Did you ever wash them like I suggested? Okay, I get up and get dressed, okay? Anyway, sarcoplasmic reticulum is smooth ER in a muscle cell, but it contains something special called calcium. And calcium is what causes a muscle to contract. All right, what else do we have? Oh, T-tubules. Now we're getting microscopic. Remember I said that we had to get that sodium deep down inside the muscle cell so it could cause calcium to be released completely within the sarcomere. Remember what a sarcomere was? The contractile unit of a muscle cell? Okay, the T-tubules are the ones that run vertical. Everything in a cell runs horizontal except for one thing, and that's a T-tubule, okay? And it goes from the top all the way down to the bottom so it can cause the gates to open to allow calcium to come out. Okay. Terminal cisterna. Okay, remember the sacs that are there, which, let me back up. The endoplasmic reticulum are kind of like sacs. They're tube-like sacs. Inside of it is calcium. The openings that allows calcium to come out and go back in, those openings are called terminal cisterna. And what you have is a T-tubule, and on each side, you have a terminal cisterna. 1, 2. Let me get this turned so you can see it. Okay? Now, when these tubes open up, calcium comes out, and calcium goes back inside. A T-tubule, a terminal cisterna, and a terminal cisterna makes a triad. Whoop, there it was. 2 terminal cisterna, or cisternae, and a T-tubule is a triad. I'm going to ask you that somehow on an exam. Here's a picture of it. Okay, I drew it for you the other day on the board, but these guys get a lot more money because they can draw better. And in the one I did on the board, it was blue. This one, it's green. This is your T-tubule, and all this blue represents endoplasmic reticulum, okay? Sarcoplasmic reticulum, because it's in a muscle cell. And here's your Z line. Remember that? Here's a Z line. There's another one kind of hidden under there. And this distance from here to here is a sarcomere. And every Z that you see is a sarcomere. So there's 1, 2, 3, 4, 5, 6, plus all these inside. Okay, so there's lots and lots and lots and lots of sarcomeres. And here's your M line. And you can see a little bit of light and dark, but don't worry about that right now. And we see the sarcolemma. This isn't the mycelium, this is the sarcolemma, okay? Which is the phospholipid bilayer. If we expanded on that, we would see the purple balls with the yellow legs, okay? Okay. Anyway, if you look real carefully right here, you can see a terminal cisterna, and you can see another one right there. And this is the T-tubule, okay? And these T-tubules are connected. That's what that goes to. Myofibrils. Okay, now we're in a muscle cell, which is a myofiber, and it has clusters of, let me back you up one. It has clusters of these. So this is a muscle cell, a myofiber, and it has clusters of these. These are myofibrils. See it right here? Myofibrils, that's what that circle is showing you. And inside the myofibrils are the myofilaments, actin and myosin. Okay, that's what I'm leading up to. Okay, myofibrils tells you what it is. Myofilaments, actin and myosin. This is where when we get to the myofilaments is when we're going to talk about the sliding filament mechanism. Okay, blah, 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 blah. 2 types. Now there is, you know that there's more proteins now because You've already seen the lecture on this, and there was titin, there was troponin, tropomyosin, and then actin and myosin. Now, when they say that there's two types of myofilaments, they're talking about the ones involved in the sliding across one another that brings the Z line together. Okay, you have a Z line on one side or Z disc on one side, a Z on the other side, and the Z's are attached to actin. So when actin is pulled by myosin, remember that fires, when it pulls the myosin, it brings the walls of closer together, which is our Z lines. Okay, thick filaments. Remember the thick ones are myosin and the thin ones are actin. Good? Thick filaments. Okay, don't worry about this, the nanometers. Don't worry about that. It's composed of myosin. That's all I want you to remember. And remember, the myosin had a head and it was cocked. ATP cocks it, yeah? And when it forms a cross bridge, remember it bends, it fires. Okay, that's what that's telling you. Cross bridges. A cross bridge you get from an actin and myosin bonding together. And I drew that for you. I think I made the actin blue and the myosin red. Thin filaments, okay, don't worry about the F and the G, actin. Okay, and that's it. So the thin filaments are actin, period. Okay, now drop down. You see tropomyosin. Remember I said now, the actin and myosin are directly involved in the sliding filament mechanism. But it has other proteins involved. Troponin, tropomyosin, titin, okay? There's troponin. Now remember, man, I gotta open up the door. It's too hot in here. I'll be right back. Oh. Door's open. Get some cool air. We're in the man cave. That's hot tamales. I got a dispenser. Okay. What else do I got in here? Got a candle. Oops. This one guy, this one instructor is just really into this proctoring. He's already sent about 25 emails. And you have to be tolerant because really I want to tell him, go get a life. Okay? But this is important to him. It isn't important to me. All right. How you guys been doing? Miss me? Kind of wish you had those lectures back on. They were fun. Okay. If you think that this requires a lot of your time, no, it doesn't. Think of the time that you traveled. Add that to the time that you're spending on a computer. Hey, you're coming out ahead. And for those of you that are not computer savvy or don't want to be computer savvy, I feel for you. Because I tell you what, I think my butt, and we know I don't have one, is so flat right now. It beats my mom's, okay? All I'm doing is sitting, typing, answering emails, doing exams. All right, I feel sorry for myself. Okay, troponin, remember that's where calcium attached to and bent the tropomyosin away from the active side of actin. And I'm gonna show you this, but that's where those two words come in. Okay, now, I kind of drew this. Mine didn't look like this, but remember this was part of a sarcomere. Let me get the arrow. This is part of a sarcomere. And what you're missing is right here is the Z line or the Z disc. And then this represents actin. The red balls represent actin. The green candy cane thing is tropomyosin, and the little blue balls on top of it, I'm trying to get this arrow. Arrow, there it is. The blue balls on it are troponin, okay? And it shows that, now what it doesn't show you is where titin is. Titin's over here. That's holding the myosin. See how thick the myosin is? And then this is your M line, and the distance from here to here is your H zone. H is in Henry zone. And when it forms a cross bridge. Now, see, it can't form a cross bridge because the tropomyosin is hiding the active site. But when it does, these fire and bring the actin in this direction. The myosin doesn't move. It just moves the actin. Good? Anyway, this is what I tried to draw for you. Okay, this one looks a lot better. And this talks about, this kind of summarizes, it tells you what a sarcolemma is, sarcoplasm, sarcoplasmic reticulum. See all this junk? Okay, those are terms you're going to need to know because I'm going to ask you questions on it. Boy, I don't know what happened there. Let me go back. Okay, so anyway, that's what that was. Next one. Thick and thin. It talks about how we get that striated appearance. And we learned this when we did the first third of the semester, when we looked through the microscope at muscle. And one of the things we looked for in skeletal muscle was the striations. And we said we had to look a lot more careful at the cardiac, but it had striations too. And then it says smooth muscle did not have striations. And I'm going to show you at the end of this video clip, I'll show you why it doesn't have striations, even though it's still set up the same way. You just can't see the striations. Anyway, the striations are caused from diminishing of light. I'm going to go back a couple of slides for you. Okay, do you see now if the light was, watch for the arrow as soon as I find it. There it is. As soon as the light comes up this way, part of the light is diminished by this, then by this, then by this, then by this, by this. By the time it gets to your eye, it's dark. Do you notice there's nothing right here? So that means the light coming right through here would go pass right through this. All it's got to go through is the myosin, and it goes right through here to your eye. So this would be light. This would be dark. Okay? Dark bands. Remember I said you have an A in dark. So the dark bands are the A bands. The light, you have an I in light. I bands are the light bands. I just showed you what they look like. And it says the I bands also contain titin. That's holding the myosin, but that's really, really narrow too. Oh, gosh. All right, now, since we have invented the electron microscope, we can see all this stuff. So we're not just hypothesizing this and telling you this is what we think it is. We actually know. And you guys are killing me here. Did you hear how many of these things I'm going through? Fob. What else did I do? All right. All right. Anyway, H zone, remember? When the actin stops and there's a gap and there's another actin on the other side, that space called H zone. It says where there's no thin filaments, which thin filaments is actin, okay? M line right down the middle. The Z disc. The Z disc or the Z band, remember that's the outside part of the sarcomere. Okay, there you go. Sarcomere is the functional contractile unit of a muscle cell. You think you're going to see that on an exam. Okay, and it says it's defined by, which means the outside borders are the Z disc. You got a Z disc on one side, you got a Z disc on the other side. That defines the outside of the sarcomere. And it says myofibrils, that's the stuff inside the muscle cell, are repeating sarcomeres. So you may have 25 or 35 or 45 sarcomeres head to toe, head to toe, head to toe in a muscle cell. Got it? Okay. It says the sarcomere shortens, the muscle contracts. Okay, that's a no-brainer. Okay, now look at what we have here. Okay, this I kind of drew. Remember I had 9 of them and I drew it with black, the Z lines. Okay, the Z lines you got to look are really small here, but they're supposed to be, as soon as I get that arrow back up here, there we go. There's supposed to be Z's here and Z's here and Z's here and Z's here. So we have a bunch of sarcomeres attached to more sarcomeres, attached to more sarcomeres, attached to more and more and more and more, and we still don't even have it completed a cell. Okay, and then so this is the Z disc. There's the M line. That little light spot in between is the H zone, and then we're back to the now. Look at the I band. This is kind of weird. You see the I band is part of this sarcomere and part of this sarcomere, because there's no myosin there. Remember, the myosin is held in place by titin, okay? So there's a titin going right here and attached to that myosin. Then there's another titin holding the myosin. So this light band is actin and titin. Good? Okay. Anyway, you don't worry about this stuff. And then we're back to showing you that one slide I showed you before. This is a sarcomere that I tried to draw for you. There's the titin holding the myosin, and there's the actin attached to the Z line. And then if you look down here, this is a photomicrograph taken with a transmitting electron microscope, and we can see here's your Z line, here's your Z line. Remember, half of the light band belongs to this sarcomere and half belongs to this sarcomere over here. Here's your M line, and you notice how it goes like this. Watch, light, really dark, sort of dark, really dark, light again. That's because the really dark is this part right here. So in other words, not only do you have myosin, but you also have actin, making it really dark. Right here, that's sort of dark, is just myosin. And then the light stuff is real thin filaments, actin and titin. Good? So it has I band, A band, H zone, so on and so forth. Okay. Now your thought might be, why are you going into such big detail about this? It's just parts, let me memorize it. Because we can prove how a muscle contracts, because we can put electrical current to that muscle and we can watch this picture change. In other words, this area right here will get very narrow, because remember, it's sliding over the top of myosin. Okay? So this remains the same, but this will get shorter. You'll see what I mean. It's coming up. Okay, this tells you about what changes. Does the A band change? Remember, A is myosin. Answer, no. Does the sarcomere change? Well, that's a no-brainer, okay? Of course, we know the sarcomere is going to change this is in length. H zone, does it change? Oh, heck yeah, because it'll diminish. You'll find out when a muscle completely contracts, it disappears. M line, does it change? No, it's always going to be in the middle. I band, that's the light. Does it change? Yes. Does the Z disc change? Now, this is careful. The Z line or the Z disc changes as opposed to the other side because they come closer together. But the actual Z does not change. It doesn't all of a sudden change form. It always remains that Z. Okay? So the answer is no. The answer is no. I ask you that. Sliding filament theory. Okay, we changed this to sliding filament mechanism. Some books call it the sliding filament theory, which means we theorize this is how it's done. But since we can actually see it on a microscope and we can see the change, it's not a theory, it is actually a mechanism. And I imagine in the future, it'll be called a law. You know how science works. Okay, they got to make sure that it's like gravity. Gravity used to be the theory of gravity, and then it was just called gravity. And then when they can prove that absolutely it always is going to be there, it was called the law of gravity. Okay. Was it Sir Isaac Newton that had the apple fall on his head? And he said, gravity. I don't remember. I wasn't there. Okay. I really wasn't there. I may look old enough, but I'm not there. Okay. That's just the hard life I've had. Okay, this is going into the H changing, the M not changing, the A band not changing, the I band changing. Just going to talk about those things. Okay, that's what I put the colors in there for. Just going over exactly what we just talked about using the picture. Okay, now I'm going to show you a couple things. Here's a relaxed muscle. Okay, we've seen this. Where's my arrow? There it is. Okay, we've seen this picture before. Titin, actin, myosin. H goes from here to here. H zone. M line is in between. This is the I band. This is the A band. Okay, so see it? Now watch it. Now what we're going to do is we're going to contract the muscle. See, this shows the muscle relax. We're going to contract and we're going to watch. I want you to watch this get more narrow, this get more narrow, and the dark part, the real dark, is going to increase because the actin is going to move underneath here. Got it? Now, did you notice that? Look what happened here. The H zone is almost gone. I'm going to put you back so you can see it again. Okay, see that zone? See that zone right there? I want you to watch that, and I want you to watch this. Here we go. Now, you see how that H zone got diminished? And this is becoming diminished. Okay? We're going to do one more time. Ooh, look at this one. Now look at the color. It's all crossed over, so we almost have same color everywhere. There's going to be a little tiny band right here, which we see a little tiny band right here, where where it's still a little bit lighter. Otherwise, all the color's the same. It isn't that it got lighter, it's just that it's all the same color because that's the light that's coming up to your eye. Okay, then remember I took a marker like this and I said, here's what causes this muscle cell to contract is the nervous system, electricity. And I said, this is called a motor end unit, okay? This and this being a muscle cell, this being a axon knob. The two of these make a motor end unit. Comes from up here, comes down the brain, and this is where the motor end is going to stop. It's going to put electricity in here, which is going to be sodium, and then this is going to contract, which we just showed you. So we call that a neuromuscular junction, okay? And I drew it for you. Muscle fiber, synaptic knob. Okay, now remember I gave you the components and I said we have a presynaptic membrane and a postsynaptic membrane. The presynaptic membrane has the synaptic vesicle in it. Remember I talked about the Golgi and making the neurotransmitter, and they put it in a synaptic vesicle. Synaptic vesicle is nothing more than the same vesicle we learned about when we talked about the cell. Only this one is in a neuron, so therefore it's called a synaptic vesicle because it contains a neurotransmitter. Got it? Synaptic knob. I used to say E.T.'s finger. Now, and then I found out from you guys, you never saw the movie E.T., so it didn't really have any basis. But E.T.'s finger was not like this. E.T.'s finger was swollen at the end. And when E.T. touched something, it put off this little glow. And to me, in my mind, when I learned about the neuromuscular junction, I said, well, that's what's happening. The electricity, the motor end unit's coming out there, and we're getting a glow, which means the chemical, the neurotransmitter chemical, is going from the presynaptic membrane to the postsynaptic membrane. Good. Synaptic vesicles. And the reason it says acetylcholine, remember on the board I wrote it wrong, and then I fixed it. Okay, acetylcholine is just a type of neurotransmitter. We have bunches of different types of neurotransmitters. I just used acetylcholine because that's the one we always see in skeletal muscle. Motor end plate, we talked about that. I just mentioned it. Synaptic knob, carrying the message from the brain to the muscle cell, the sarcolemma. Remember, the sarcolemma is the plasma membrane of muscle cell. Synaptic cleft is the area between the presynaptic membrane and the postsynaptic membrane. That area is called a synaptic cleft. And then on the postsynaptic membrane, you have receptors, which are guards to the gate. And when the neurotransmitter lands on those guards, the guards open the gate and allow sodium to come in, and it'll go down the T-tubule. Acetylcholinesterase. Now, when you see that E at the end, there we go. When you see that E, it means enzyme. You have to have an enzyme to break down acetylcholine, otherwise the gates stay open. If the gates stay open, sodium continues to go down there, and the muscle stays contracted because it's going to leave calcium out. So you have to undo all of that. And in order to undo all that, you got to start with the enzyme. You got to break down that acetylcholine so the gates close, so sodium is no longer going to affect the release of calcium. Good? Okay, here's a picture. This is kind of an illustration. This is your axon knob. This is E.T.'s finger, It's not actually touching. There's a space there called a synaptic cleft. Here's your axon. You notice that because of the myelin sheath. These are the telodendria, and the telodendria they have the axon knobs at the end. Okay. And then this would be amplified. Now, remember I showed you the model of a sarcolemma, part of the sarcomere, and you have a handout on that. And this, and it shows it being straight, and then it shows it being corrugated right here. It's corrugated all the way around. They're just emphasizing this. So there's actually a space between the blue and this white you see down here, and that's the synaptic cleft. If we magnified it, this is what you would see. And then these are the synaptic vesicles, and the little green dots inside are the neurotransmitter. And that's what's actually, when this is released, it comes out, goes through here, goes across the cleft, and these are the gates. And the gates are guarded. And these neurotransmitters tell the guards that it's okay, and they open up the gate, and sodium comes in. And then sodium travels down here, and what they're missing here is the T-tubule. Okay, and this is a photomicrograph. This is, in fact, this is a light microscope showing that this is E.T.'s finger. This and this are the same thing. Pretty good, huh? It tells you about it, binds to it. This just tells you that it's opening the gate, and we have to have an enzyme to break it down. Okay, remember the calcium, which you see in this one. Calcium ions bind to troponin. What caused the calcium to come out? Sodium. What caused the sodium to go in? The opening of the gates. What opened the gates? The neurotransmitter. Where'd the neurotransmitter come from? It came from the synaptic cleft. Where was it before that? It was in the synaptic vesicle. Where did the synaptic vesicle get it? From the Golgi. Where did the Golgi get it? From a ribosome. Get how it works? Okay, okay. Okay, then it talks about the myosin and actin. This number 5 says myosin heads bind to actin, forming cross bridges. And then you know what it fires, right? And ATP is necessary for relaxation. So in order to undo all of this, that's when you use ATP. Okay, now this picture is pretty cool because this shows the sarcolemma. See how it's corrugated? This shows the synaptic cleft. This shows the vesicles being broken open, releasing neurotransmitter. It shows the neurotransmitter. Well, it doesn't. It should show one more thing. It should show the neurotransmitter. I think I have it coming up in another slide. Opening up the gates, or allowing the gates to be opened. Okay, and then this is the T-tubule, the transverse tubule, and this is the sarcoplasmic reticulum. And then the little holes that you see, which aren't shown very good, are the terminal cisterna. And these little yellow dots in here is not sodium, it's calcium. See, and calcium's coming out here. Where do you think that calcium's going to go to? Come on, think. Where do you think that calcium's going to go to? Come on, I'm not going to tell you. Yeah, I will. It's going to go to the troponin. There it is. See the calcium? That calcium right there, that dot, is that dot right here, and it shows the arrow. It's going to attach to troponin. When it does, it bends it. Now you look in here, and you could probably see just a little bit of a dark spot. Now the dark spot is opened up because the troponin has moved the tropomyosin away, so the actin and the myosin can bond. When we bond, we call that a cross bridge, and it fires. Okay. Motor unit, the brain and the nervous system and the muscular system. All or nothing, don't worry about that. Don't worry about that. Okay, remember we talked about twitches? We said it's a single contraction of a sarcomere, of a myofiber muscle cell. And we said that we're going to recognize 2 of them, fast and slow. Do you remember that? Fast was white, slow was red. And then there's an intermediate, but we ain't gonna worry about that. So would be a muscle. This would be a muscle consisting of muscle cells. And here's the motor end unit, the nervous system attaching to it. And some of them are white and some are red. In humans, pretty much they're all red. Remember we talked about the duck and the chicken. Muscle tone. remember I've said tonia, the suffix tonia meant the muscle cell was slightly constricted, and that gives you muscle tone. Okay. And then we said, just a minute, trophy is muscle size, and plasia is muscle tissue. And you put a hyper and a hypo in front of it, tells you you're getting more or less. All right, that's what's coming. Okay, then remember we talked about isometric contraction. Isometric meant you cannot overcome the resistance. Okay, picture yourself trying to pick up 500 pounds. You can't do it. Even though your muscles are constricting their poor little hearts out, you cannot flex and bring that thing up, that 500 pounds. If you think you can, you're lying to yourself. Anyway, when a muscle contraction where you cannot overcome the resistance is called an isometric contraction versus isotonic contraction. Isotonic, you do overcome the resistance. In other words, you have enough power to move that. Okay, that's an isotonic contraction. And there are two types. One when the muscle shortens and one when the muscle lengthens. Shortening is called concentric, and lengthening is called eccentric. Remember that? In case you didn't, there they are. Good? Yes, I'm going to ask you that test question. Time for another hot tamale. Probably why I'm gaining weight. Okay, here's Mama Lee. Mama Lee is going to show you that she can't lift 80 pounds. Isometric. She's trying. That muscle is contracting, man. She just cannot overcome the resistance. Okay, so she drops down to 10 pounds. This will probably be a struggle for some of you, and she can overcome it. This is an isotonic contraction. The biceps muscle is concentric. The triceps muscle is eccentric. It's getting longer. Okay, it's changing in length. Here's slow and fast. Don't worry about this. Remember the white and the red? I'm going to ask you about white and red. Which one's fast, which one's slow? That's more white and red. This shows you that it's really there. So here, this would be white. This would be red, and this is intermediate. Okay, so blend of the two. Big deal. I tried to swallow that hot tamale too quick. I didn't chew it enough. Maybe I should spit it back up, chew it again like a cow. Anyway, this is just more of that same stuff. Don't worry about it. Okay, now we're talking about how do we name, how are muscle cells, fibers, myofibers, arranged? Okay, first thing, they can be circular, like orbicularis oris, around your eye. It's round around your eye. Okay, it's round around your lips. So you can have circular, you can have parallel, rectus abdominis, rectus femoris down your, I'm touching my leg, okay? The part of the quadriceps right in the front, okay? That's rectus femoris. So they can be parallel. They can be convergent. That's like your pectoralis. It starts here, and when it moves over to your humerus, it forms kind of like a triangle. That's called convergent. And then pinnate. Pinnate means small, large, small. Like we talked about a shape of a fish or a bird or an airplane. That's pinnate. And that's what we see in our biceps muscle. Okay? And you can have unipinnate, which means one tendon on one end and one tendon on another end. That's unipinnate. You can have bi, which is 1 and 2, or you can have multi. Okay? Okay. And here's an example. Orbicularis oris. Okay, your lips. Rectus femoris, or rectus abdominis. Okay, that's the six-pack muscle. Here's the pectoralis major, the triangle, you see what I mean? And here's uni, bi, and multi. Tri would be a multi. Okay, here's those terms we used. Remember we said atrophy applies to anything when it gets small, even though it should be just the muscle tension, the cell tension. But atrophy has subcategories, and that is hypotonia, hypoplasia, hypotrophy, okay, all those things. And here's hypertrophy, to increase in size. Atrophy, general umbrella. Whenever you put a hyper or hypo out here, it says you're either going to get bigger or muscle, a bigger or smaller cell size. Oops, I didn't do the other ones for you. But there's hypotrophy, there's hypertonia, which means the cell just gets more rigid, and hypotonia, it's more flaccid. And then there's hyperplasia, more mass, okay? And hypoplasia, less mass. Mass means number of cells. Okay, here's their fulcrums and levers, okay? You might pronounce it levers. We talked about that. You're not going to be responsible for that. And this is showing you what they mean. And this is what I was showing you on if this was the biceps muscle and it went all the way down to your wrist, it wouldn't take as much energy in order to move your wrist and muscle, because you have the fulcrum is at a different point. Okay, and that's what that's showing you. It's like a wheelbarrow. I'd say some of you wouldn't know how a wheelbarrow works. Okay, but if you put all the weight back here, it's too heavy. Put the weight in the front, it's real light. Okay, so if you want to learn how to work a wheelbarrow, let me know. I have plenty of work out here for you. Okay, remember we talked about the mover is called an agonist. The muscle that opposes it is an antagonist. Okay, and then they switch. Okay, you're not always going to be an agonist. You're only an agonist if you're constricting. You're only an antagonist if you're stretching. Okay, and then we had the helper, the synergist. Remember that? Okay. Naming the skeletal muscles. Action, flexion, extension, one way. Specific body regions, abdomen, transverse abdominis, external oblique abdominis. No, that's not right. External oblique, internal oblique. I don't think there's an abdominis after it. Anyway, rectus femoris was okay. So it depends on where it's at. Muscle attachments. I like this one. Sternocleidomastoideus. Sternocleido, clavicle, mastoideus. Okay, the big long name shows where it's attached to. And orientation, orbicularis oris. Okay, orientation, it's round. Size and shape, magnus, gluteus. Okay, you got, some people got a big gluteus. Okay, gluteus maximus. Okay, the shape. I know the name of this. I'll think of it in a minute. The example I usually use. Where the heads or the tendons of origin will sometimes do it. Okay, here's a whole list. Here's a whole list. Man, it's driving me crazy. All right. 3 types of muscle in the human body. We started with this, huh? Skeletal cardiac muscle. You know the differences, da, 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 da. Now, that's it for skeletal muscle. I have finished it. Now what I want to do is I want to talk to you about what's the difference with cardiac? Nothing, except for we have the intercalated disc and they're not lined up nice and parallel, but they're still striated, still the same setup, still actin and myosin and titin and all that other stuff. Okay, it's striated. Remember, it says Y-shaped. Remember, sometimes they were branched out like that. I showed you. Intercalated discs. Okay, gap junctions, desmosomes, autorhythmia. Remember, I just threw this in here because remember the heart is autorhythmic. We went into the heartbeat and all that kind of stuff, and we know that there's, after the Purkinje fibers, it loops up to the sinoatrial node known as the pacemaker, a little loop like that, yeah? And it's involuntary. That's pretty good because sometimes we forget to think, and if we forget to think, our heart doesn't move. Okay, that means we'd be dead. So we give that to the involuntary part of the brain, so we don't have to worry about autonomic. Okay, here shows essentially the same thing. Okay, it just shows these are the gaps. These are not the Z lines. The Z lines are over in here, but it shows the same thing. Smooth muscle. Okay, smooth muscle does not have striations. Is it still set up the same way? And the answer is yes. The reason you don't see the striations is everything in smooth muscle is not parallel like it is in cardiac and skeletal muscle. It goes on 90 degrees, 45 degrees. So smooth muscle has the ability to constrict and to constrict this way. So in order to constrict this way, they have to be coming at an angle. In order to constrict this way, they have to be parallel. That's why you cannot see it. I'm going to show you a picture. Remember the cells are fusiform? Remember what that was? Small, large, small, small, large, small, like an airplane. No striations, just told you why. Involuntary again, you don't have to worry about it. Here's what I mean. If this was skeletal or cardiac, actin and myosin would all be parallel. As you can see, this one's parallel, this one's going up at a, at about a 60 degree. Okay, so this means the cells can not only constrict this way, which is what you see over here, they got shorter, but they can also constrict this way. Got it? Development, you don't need to know. Shows the comparison of the three, top one being skeletal, middle one being cardiac, lower one being smooth, where you can find it. Still made up of the same things. It's saying we're still made up of a muscle, a myofiber, a myofibrils inside. Okay, it's just the orientation of those. That's it. We're going to end this and I'll upload this and this will be part 2. Didn't do what I told it to do. Okay, hang tight. Nope. Nope. Okay, I got to go down to here. Stop recording. Here we go.